Okay, let's start. So hello everyone, good morning, good afternoon, wherever you are in the world. Thank you for joining our webinar today. My name is Thomas von Larcher. I'm Managing Editor Engineering for SN Applied Sciences, and I'm helping to run the webinar today. Today we have Dr. Shogo Hamada from Tohoku University, Japan, with us. Dr. Hamada received his PhD from the Tokyo Institute of Technology and was assistant professor at Tohoku University. He spent also some time in the US and was postdoctoral fellow at the Kavli Institute at Cornell for nanoscale science and research associate at Cornell University. Recently, he returned to Japan and is now lecturer in the Department of Robotics at Tohoku University. Dr. Hamada's research interest lies at the intersection between robotics and nanobioengineering. And he currently hosts a topical collection with the journal SN Applied Sciences called Molecular Robotics, and his talk today is also about molecular robotics. So before we start with the talk, please make sure you are muted. And after the talk, we have time for questions. So please type your questions in the chat box as we go, or after the talk, you can unmute yourself and ask questions. So with this, Shogo, we are delighted to have you with us today, and the floor is yours.
Thank you very much. So then I'll share my screen. Wait a second. Can you see my screen now?
Yes.
Perfect. Okay, let me start. So, hello everyone. My name is Shogo Hamada. I'm working on a new kind of machines and robots built from biomolecules. And before I start, I would really like to thank Thomas for kindly introducing me and also giving me this great opportunity to talk about my latest research and sharing this excitement of this new frontier called molecular robotics. So, well, actually, the Thomas already introduced me about my background, but this might be not a kind of like a repetition again. But let me briefly talk about my background. So actually, I was originally started from control and system engineering, building mechanical robots when I was in college. But I started to envision building like new kinds of robots with new materials. And the new material basically leads to new hardware, right? Thus new capabilities, which we're not able to implement by conventional mechanical systems. So that was the main reason I jumped into this new field called DNA nanotechnology, which eventually led to molecular robotics, today's main topic. And as Thomas mentioned, I recently moved back to Japan, my home country, from the US as a lecturer here at the Tohoku University. And I also joined the Molecular Cybernetics project. It's the government-funded project here in Japan as well. And previously, I was at Cornell, and today's talk is mainly from the works I did at Cornell with my colleagues. And before moving on, I like to mention like several Springer Nature-related works. And I'm first, as Thomas mentioned, I'm currently working as a guest editor of Molecular Robotics section in Springer Nature Applied Sciences. And I'm also working as a section editor of the Molecular, Nano, and Micro Robotics in the Encyclopedia of Robotics as well. So please feel free to take a look, and also, feel free to contact me if you're interested in those for further details. So let's jump into today's topic. So my mission is to create robots with characteristics of life. So from the nanoscale, using biomolecules. In short, creating lifelike robots. And mimicking characteristics of life by machines itself is not a new concept, actually. So we can even trace back the idea back to like a Greek myth, actually. And for instance, this figure from 1980s explained the concept of self-replicating robots in a mechanical engineering fashion. So in a sense, creating such lifelike robots, including like a self-replicating and evolving machines, are one of the fundamental, but also still an ultimate goals in robotics. And right now, roboticists approach to realize this dream, for instance, by building modular robots based on mechatronics, for instance. So these examples, are truly fascinating. But at the same time, we can also see some fundamental obstacles towards this goal. So I was one of the students in robotics, actually, who was intrigued by those examples. But at the same time, the more I learned, I started to have mainly two questions like this. The first question is about the material. So who is going to prepare those complicated parts from the raw material level? That was the first question. Another question was about the scale in terms of numbers and sizes. So it is difficult to build billions or, of complex units in reality, right? And also there are some kind of limits in miniaturization if we rely on simply with the mechatronic components. So how to solve this question? That was my original question I had. And in our case, we return to the source of our inspiration. So it is too complicated to overcome in mechatronics. So how about building robots by bio? That was the fundamental motivation behind here. So of course, our life is a system, right? Built from biomolecules and biochemical reactions. So if we can build such robots, that could easily overcome the limitation of current robotics and may achieve lifelike characteristics. And as shown in these beautiful examples, researchers are now trying to push these boundaries by bio-enabled approaches like tissue engineering, cyborg biorobots, or synthetic biology. So, but still, the current approaches themselves have its own limitation. So all of them are still relying on the current architecture of life, like cells or like tissues. So I wanted to push this limit one step further. So something really big needs to start from something really small in this case, smaller than the cells. So as a kind of alternative approach, can we build a robot from scratch, from the molecular level, without the black box of life? That led to the idea of molecular robotics. So I recently created this figure as a part of the encyclopedia, actually, to explain the historical background of the field. So basically, the current molecular robotics has two types of background. One has a roots in chemistry, emphasizing more on a synthesis of new molecules that can achieve like state transition, for instance. And the other has more bioengineering background, emphasizing to create systems by combining various kinds of biomolecules. So today's topic is related to this new trend of biomolecular robots. So how are we going to approach this? So as we all know, a robot has three primitives, right? Sense, plan, and act. And hardware-wise, each corresponds to like sensor, circuits, and actuator, in addition to the structure of the robot itself. And molecular robotics applies this analogy that we are already familiar with. And of course, instead of using mechanical parts, we can create four elements all by molecules, biomolecules in this case. All devices can be actually constructed and also integrated into one systems by biomolecules, especially by using DNA. So for instance, we can create any kind of shapes at the nanoscale with DNA, and also we can build logic gates and even like neural network by DNA as well. And we can integrate them. So DNA is a kind of hub molecule of biomolecular robots. And DNA plays a central role in molecular robots due to several reasons, actually. Since the time is limited, so I'm not going into details today, but in general like this. In addition to the genetic material, as you may know, DNA has unmatched potential properties as a generic biomaterial. The first property is the construction. We can construct varieties of structures across scales by self-assembly, like a nanoscale Lego block that can form by itself. And in case of scales, in terms of sizes, we can build from nanoscale structures to like a micro scale structure. In terms of numbers, since we're dealing with molecules, we are easily can get like Avogadro's numbers of molecules and create robots from there. So from that sense, we can easily overcome the current limitations of mechatronics. And second is about this control. We can easily control this behavior by sequence design and enzymatic reactions. So the formation is predictable by simulations. And also we can use enzymes to cut and duplicate, basically manipulate DNA. And third, we can connect varieties of molecules by the help of chemistry. So by using all of these properties, we can create like various applications by bridging nano and bio worlds from materials and healthcare which can lead to the new application in robotics as well. So now both tools and design methods are ready to build systems using DNA. So the creating a robotic system from the nanoscale using DNA is probably a next frontier in robotics. In fact, researchers all over the world started to develop various types of molecular robots using DNA and with other types of biomolecules. We are witnessing a kind of like a Cambrian explosion of biomolecular robots recently. From like a DNA spider, which can, like a DNA walker, autonomously walking on a nanoscale substrate, or like assembly line, or like autonomous sorter, or like by using the electrical field, we can create a nanomanipulator, or even we can create like a DNA nanocage that can be used as a kind of future drug delivery devices, for instance. And also for the hybrid materials, if we combine with motor proteins, we can already create like an amoeba-like robot or like a swarm robot by using these biomolecules. So most importantly, these state-of-the-art robots exemplify that this approach is feasible enough to create systems from the molecular level. So the molecular robotics allow us to overcome limitation of mechatronics and even opened up the route to create the lifelike robots based on these molecules from the molecular level. So now is the time to realize these lifelike characteristics based on this approach. So I've been working on this field, mainly focusing on to build various types of unique DNA-based systems across scales, like from nanoscale to macroscale, as a kind of foundation of biomolecular robots. At the nanoscale, I created the new type of junction, and based on that junction, we created a DNA nanostructure platform, and then we created the quasi-self-replicating system with our colleagues. And also, by using the technology called DNA origami, we created a self-reconfiguring nanostructure with our students as well. At the microscale, I did mainly about the DNA hydrogel-based system, especially focusing on enzyme-based fabrication of those hydrogels. And that can allow like an interaction with human at the bulk scale. Basically, you can really touch the DNA. So those hydrogels are all made from DNA. And also we implement like a controllable triggered biofunctionality, including like a protein expression, for instance. So those inventions at both nanoscale design and microscale fabrication enabled the lifelike machines at the mesoscale, which we call artificial metabolism. And as a first step towards this goal of creating lifelike robots, we targeted to create artificial metabolism. So among various characteristics of life, metabolism is actually a key mechanism that can make life alive. So the combination of anabolism, generation part, and also the catabolism, degeneration, sustains life as by a continuous flux of matter and energy. So, this is a kind of like a fundamental engine behind dynamism of life's materials. And also, it is a basis of further lifelike characteristics. So in short, we eat food and use it as an ingredient to create and maintain bodies, right? And also at the same time, we decompose unnecessary materials. That's what we are doing, basically renewing our body. So as Prigogine coined the term, like life is indeed a dissipative structure, right? So the artificial metabolism is also the key to overcome limitations of current robotics. For instance, like a production from a raw material level. And also we can achieve like a continuous and scalable synthesis and assembly. And also it does not rely on existing architecture of life if we can achieve this. So what if we could mimic these characteristics, right? This will lead to lifelike molecular machines or robots. That means robots that can eat, grow, decay, and probably more. And for the first time, we made that kind of system. So of course, the metabolism in life is extremely sophisticated, as we can obviously see in this example of human metabolic pathways, for instance. So we needed to start from really simple, something super simple. And we made something like this. What we call is the DASH system. So the DASH system is our first successful attempt of implementing this concept of artificial metabolism. DASH stands for DNA-based assembly and synthesis of hierarchical materials, which combines like 2 cycles, which has the biochemical synthesis, decomposition, and also the dynamic and dissipative assembly. So it's really simple, as simple as the coupling like only 2 cycles actually. And by using this system, we can dynamically and autonomously create hierarchical material like life in a more super simplified way, of course. But starting from like a building blocks, DNTPs, which can lead to like a polymers, DNA, and then which can form into like network hydrogels, and then shapes and pattern structures. So based on this system, we can build machines and robots. And I'm going to talk about a little bit more details about this. So the actual implementation of this dash system is extremely simple. So for the synthesis side, we prepare the seed structure and building blocks, DNTP, and of course the enzyme to synthesize DNA, and which creates this long DNA from there. And on the other hand, assembly side, we use the microfluidic device with pillars inside there. And basically pillars mix the solution and dynamically assemble synthesized DNA into those patterns. And the key here is both are simultaneous and autonomous. So there's no human intervention during the whole process. So only the constant flow is here. So basically what we are doing is basically flowing in this synthesis solution into this microfluidic device. And that's it. And as a result, we can create the spatial temporal control of pattern generation. Let me show you one example. So in this crosshatch designs case, this is a microfluidic device we made, and the flow is coming from the right-hand side. And we have a bunch of pillars inside this microfluidic device. And if you run that solution, within like 2 hours, we can see this kind of like a crosshatch patterns. So I would like to mention and emphasize that this millimeter scale patterns contains like millions of DNA inside, synthesized and then assembled by the system by itself. And of course, not only the crosshatch patterns, from like 1D lines to 2D patterns, and even like various kind of shapes, we can easily design the geometry by placing the pillars based on certain rules and the CFD, computational fluid dynamics simulation. So that was the generation side. So then let's integrate, anabolic and then catabolic pathways, right? Not only by building, but we need to decompose the object we created, right? But the key here is that we need to integrate both in an orderly fashion, right? So in addition to the generation solution, which contains like enzymatic synthesis and assembly, we need to add like a degeneration side, which can do like a dissipation and enzymatic digestion, basically cutting DNA into pieces. And the key here is, of course, we cannot simply, mix both at the same time, right? So we somehow need to coordinate the order by switching those 2 pathways. So in order to do that, we implemented this autonomous built-in feedback using the generated structure. So as a result, we can do the generation, then degeneration, and then return to the original state like that. And let me explain a little bit more detail. So as you can see from this simulation result, we have like 3 types of solution running in. So the black side is the generation side, and the red side is the degeneration side. First, laminar flow creates dash patterns at the center of the device because there is no like intervention between red and blue, red and green, red and black, sorry. So the black side can start creating dash patterns. However, once the patterns are generated, then the pattern itself can alter the flow inside. Basically, you can mix both black and red. So it triggers a kind of mixture generation degeneration solution. And as a result, this feedback creates the, switches the pathway to the degeneration side. So here's the actual experimental results. So here we have like a degeneration, generation, also degeneration. 3 solutions are running in. And first, you can see the patterns are generated at the center of the device due to this laminar flow. However, once the patterns grow further, as a result, this structure itself started to alter the flow by mixing 2. And as a result, the whole pattern is going to be degenerated. And this autonomous and cyclic regeneration process can be repeated up to like 2 cycles right now. And importantly, the key here is that now we can describe the behavior I explained, analogous to the mechanical robots basically by simplifying it, and can program novel class of machines by using this abstraction. So basically, we can regard this, this behavior with like a 3 states with state transition by using finite state automata, with the initial state and then growth state switch to like a decay state like this. And these can be these have a state transition like this. And the strategy to program this behavior is that we can treat this FSA as a kind of unit and connect them by the signals, what we call like a flow altered signals here. And we have 2 programmable parameters here in order to create the behavior. One is the different waiting time, Tn, until it switches from init state to the growth state. And the second parameter is how to connect these flow altered signals between FSA. And we have like 2 programmable parameters here. And based on that, we actually designed 2 types of machines here. One is locomotion and another is racing. So I'll talk you details about that. And these programs are actually inspired by 2 examples in biology. So the first inspiration was the behavior of this kind of cellular slime mold. So we set up goal to mimic this kind of overall behavior of locomotion. But of course, the mechanism itself is totally different. Another inspiration is this actin filament. In order to implement this behavior, we're inspired by this, another example in nature. So the spontaneous assembly of actin molecules happen at one side, and the disassembly happens at the other side, creating this kind of what we call treadmilling behavior. So our DASH system can implement similar, directional process by the dynamic formation DNA network, in addition to the synthesis and decomposition of DNA molecules themselves, all powered by artificial metabolism. So inspired by those examples, first we designed our locomotion behavior as a polarized regeneration process like this. Basically, the head side of the body always generates this new body, and the tail side degenerate. So as a result, it looks like it's moving. So we can program that kind of behavior as a kind of serial connection of FSA with one-way signal. So these M1 to M6 represents each FSA unit, which I explained before. And we have like a different, like a waiting time of the timer from T1 to T6. So as a result, this M1 start forming these patterns first, then M2, then M3, then M4. However, once they start forming, it will automatically switch to the decay behavior. And as a result, the degeneration also starts from M1 and then M2 and M3 and so on and so on. So we can create those kind of behavior by combining those FSA units. So the signal propagation, this one, also ensures the complete transition to the decay state at the downstream as well. And based on this program, we can interpret this idea to the actual microfluidic device. So each FSA unit represents one region, and different time, waiting time, represents the different gap width inside this microfluidic device. And this direction of signal propagation is represented as a flow direction. So in this case, flow coming from right side to left, like this. And as a result, we can create like a moving body against the flow. So here's an actual examples. So from left to right, you can see the machines are moving. And of course, in this actual, in this video, we speed up the display speed, but the actual speed is like a 2.3 millimeters per hour. So it is a relatively slow robot in a sense. And of course, we can create like a multiple geometries, not only like a straight tracks, but also like a curved tracks, and also like a different width, etc., by using this method. So that was the locomotion. So the second machine, the racing behavior, was realized by expanding the locomotion program. In particular, the program itself is basically expanding the locomotion program into like 2 parallel tracks instead of 1. And the key here is that they introduce this interference between 2 by flow altered signal as a kind of like a simple interaction between 2 bodies. And the interpretation of this program is also simple, like this. So 2 tracks without any like a physical boundary. It's going to be like an actual implementation. And as a result, we can create a behavior that once the one side releases the race, then that body slows down the other by triggering the state transition to decay by this flow outward signal. So as a result, once the one releases the race, the body slows down the other, so we can see the difference between those 2 bodies. So I believe seeing is believing. So let me show you the video. So here we have 2 tracks, track 1 and track 2, and both machines will start from left side and then go to the right. And again, the flow is coming from the right side, but the machines are moving left to right against the flow. So first, both bodies are becoming generated like this, and then locomotion starts. And of course, first, both robots moving at the same speed, but due to the random factors in the system, the symmetry between those 2 bodies were broken like this. So once it happens, then in this case, body 2 leads the race and it slows down body 1 like this. And the interesting thing here is, the body 1 is starting to degenerate by this interference by body 2. And as a result, we have the complete degeneration of body 1 as well. So in a sense, like a sophisticated lifelike behavior, like by a combination of these, like interaction and also racing, and also certain kind of like a simple selection can be realized by this artificial metabolism. So the DASH system could be used in various applications. Here are some examples. So the key characteristics of the system is the material regeneration, right? And here you use this generation part, only the anabolism part for the pathogen DNA RNA detection. And this characteristic of the DASH system is utilized in both like amplification and readout part of the detection. So we prepared a generation seed that can be amplified only if it matches the target DNA or RNA. So that means we can basically create like a simple if-then rule. Only if you have the target pathogen, then you can see those DASH patterns generated. If there's no like a target pathogens, there's no synthesis, so there's no generation of patterns. And also the point here, another point here is that the generated patterns itself allow really easy readout. So we can create like a mesoscale patterns, like a sub-millimeter scale patterns from here, right? So it could be observed by microscope or even by like a smartphone camera, for instance. So we utilize this self-generating ability for the amplification readout, and we can use this as a kind of pathogen detection for RNA and DNA. We also demonstrated the use of this DASH material beyond DNA by creating like a hybrid materials by attaching various types of material like proteins or quantum dots or like even gold nanoparticles from like a biomolecules to even like a non-bio materials like this. Furthermore, we also demonstrated that the DASH material is still retaining the characteristic of DNA as a genetic material by expressing proteins like this, in this case, GFP. So this demonstration could open up a route to producing more useful proteins, including like an enzyme from DASH, for instance. So this DASH system itself is still a kind of like a simple system. However, we can see that there are multiple applications future from healthcare to materials. So finally, I would like to briefly mention about our future perspectives. So we are trying to expand this system based on this artificial metabolism and implement more lifelike characteristics to machines, which leads to like a self-replication and evolution actually. So for instance, for the replication, artificial metabolism already achieves this kind of renewal of the body, right? In case of DASH, this preliminary results show the kind of division of this body into 2. I'll skip the details, but we are at the kind of like a start of this kind of new level of the lifelike characteristics now. And for the evolution, as I briefly mentioned, genes can be implemented to DASH, right? So it may be able to produce enzymes that in turn generates the next generation of the DASH. So by using this, we may be able to create a certain kind of a cycle, which may lead to this evolution. So we just started, right? We are only at the beginning of the implementing such new capabilities of the robots. But I believe from these actual examples, we just started to create, I believe that the dawn of this lifelike molecular robots is here. Last but not least, I would really like to thank the project team and also the assistance from the students and also the collaborators and also the funding agencies. And thank you very much for your attention.
Thank you very much, Shogo, for this very interesting and nice talk. Excellent.
Thank you very much.
So now we have time for questions. I don't see questions in the chat box right now, but if you would like to ask a question, please unmute yourself or type your question in the chat box now. Shogo, I noticed that this topic is multidisciplinary, of course. I mean, it's chemistry, biology, mechatronics, engineering, etc. And it's also across scales, right? So I can imagine there are challenges and limitations. Can you tell me a bit about the current limitations? I mean, I noticed a few years ago there was a discussion about what was it, sticky finger and fat finger discussion, right? So this was about limitations and future perspectives. So can you explain a bit more about this?
Yes. So that kind of like a sticking fingers problem was that the discussion, we had in, I believe, in 1990s and 2000, I believe. So the problem was that it is, simply speaking, it is really hard to create to manipulate molecules by molecules, right? That was the fundamental, problem. And so let me go back to my slide, for instance. Let me share my screen again. Okay, can you see my screen now?
Yes, we can see.
So those fat and small fingers debate was done by Drexler and Smalley and in this 2001 here. And there are two approaches basically in order to solve this problem. So one is using chemistry. We can synthesize, molecules by chemistry to avoid this kind of manipulation of one atom by atom manipulation, for instance. And another approach is using bioengineering. So bioengineering can achieve like a synthesis of molecules by using enzymes, for instance, right? So it's not like a manipulating atom by atom in a sense, but we can still create those structures based on a more systematic way. So that actually, that discussion and nanotechnology actually was a kind of one of the reasons that the researchers started to think about the like this kind of approach by using chemistry and nanobiotechnology. And actually that led to this kind of like a new field of molecular robotics right now.
I see. Okay. Yeah, I see. Thank you. And talking about real-world applications, you already mentioned healthcare materials and also artificial life. And I mean, healthcare, I think most interesting is diagnostics and therapeutics as well. So can I say the molecular robots talk to cells and other biomolecules in my body or so? Is that what we have to expect in future times?
Yes, definitely. In a longer future, I would say, longer term goal, I believe. So still, there are multiple obstacles. If we try to put those DNA robots directly inside our bodies, obviously. However, theoretically speaking, if we use biomolecules, it will allow us to communicate with other types of biomolecules inside our body, right? So actually, the some examples, like this DNA nanocage, started to develop those kind of, molecular robot. And so in this example, they tried to create a robot that can work inside cockroaches, for instance. So they tried those kind of experiments. So the researchers have been starting to try those kind of like, communicating the molecules inside like a living systems right now.
Okay. Interesting. And this will affect the society, so to speak, right? So and especially we as publisher, we are interested in fulfilling the ethics guidelines. So I can imagine this would cause some ethical discussions or so. Are the researchers aware of such ethics guidelines or discussions, or is ethics a topic for researchers in robotics and molecular robotics in particular?
Yes, so actually, this is a good question. So the researchers in molecular robotics are obviously aware of this potential, ethical concerns. Of course, we are still in a preliminary, research. So it might be really far to the actual, implementation of those kind of stuff. However, we already started those kind of discussions, actually. At least for instance, in Japan, the molecular robotics, researchers created a kind of guideline for those kind of ethical concerns. And we are having those kind of discussions between researchers right now.
Okay, well, that's good to know, good to hear. Okay, so I don't see any questions in the chat box, and yeah, apparently there is no question right now. So then we will close the webinar for today. Shogo, thanks so much for your very interesting talk. It was very nice. And again, to all, thank you for joining our webinar today. And have a nice day or a nice evening, wherever you are. So see you and bye-bye.
Thank you very much. Bye.